Well, I'd like to thank Morton and the organizers for the chance to talk to you today, and thank everybody for showing up to this. I know there's people from all over the world, and it's all hours of the day. It's 7:45 a.m. here, and I know that in Japan it's way past my bedtime. So thanks everybody for showing up. And I'd like to talk to you today about some work we've been doing on environmental microbes. I think of it as beaches to the bench, and today I'll talk about microbes that degrade carrageenan that are isolated from the local waters. And so for those of you who don't know where I am in British Columbia. I'm actually on Vancouver Island here. So this is the far west coast of Canada. UVic is located on the tip of this island, and so we're surrounded by water on three sides. And this is an aerial view of the university with my lab tucked in there, and you can see the ocean way in the background. And then we have, hang on here, I got to get some stuff out of the way. There we go. And way in the background here, you can see Mount Baker. This is Sunset in Victoria, and so this is actually a mountain in Washington State, down in the U.S. So we're a marine environment, and so it's perhaps not surprising that we see a lot of this kind of thing after various storms, winter storms, spring storms, or whatever. So here's a local beach where a kelp bed has been ripped up and thrown up onto the shore. Here's sea lettuce that's been thrown up onto the shore. And when you're walking around, and eventually this stuff disappears, and it's easy to ask the question of where does it go? And part of the answer to that is the mechanical action of waves and tides and things like that, which break this into smaller pieces. But really, where does that photosynthetically fixed carbon go? And that's where the microbes come in, that they actually will break down this stuff and recycle that carbon. And because this biomass is 50 to 60% carbohydrate by its dry weight, this recycling by microbes becomes a glycobiology problem. And so that's where people like me come in. And why is this self-interesting other than just the locale? It tends to be kind of topical for someone like me. But it's also interesting because the sugars that tend to be in these algae, these marine algae, are what I call weird. And so here's an example of three actually related polysaccharides from red algae. And what I'd like you to note is how much L sugar there is in there. So there's two of these are about 50% L sugar, which is unusual. L sugars are comparatively rare in terrestrial polysaccharides. We also have these 3,6 anhydro bridges, which makes us kind of a biochemically weird sugar. And we also have a preponderance of sulfate. Now, these aren't particularly sulfated polysaccharides. You're going to see in a bit much more sulfated polysaccharide. But our overriding question with all of this is, how do the microbes that recycle these polysaccharides deal with these weird sugars? It's an interesting biochemical problem. Now, of course, there is some relevance to it. As everybody knows, that the vast majority of photosynthetically fixed carbon is present in land plants, and comparatively less in the oceans. But the rate of recycling is orders of magnitude higher in the ocean than it is on land. So it's actually a really important part of the global carbon cycle. So there's this interesting aspect to it. Of course, you can also try and throw in the applied parts of it, where you could take that biomass, convert it to sugars, and then ultimately convert it to other useful things such as ethanol, but not necessarily ethanol. And there's a number of really interesting studies that have actually done this kind of thing. So this is a potentially interesting source of renewable energy. Now, this all stems from this idea that this algae, this biomass, has broken down into these bits and pieces. And of course, the biocatalysts are the important parts of this. And these obviously originate from the marine isolates. So in terms of being able to study them, we're really interested in the marine isolates and the genomes of these. Sometimes they're available. When we started this, there weren't many available. Now there's more. And if you have the genome, you can annotate the K-zone. This gives you an idea of what the carbohydrate active enzymes are, and you can predict the biocatalysts. Then you can put these under the microscope, and you can do a variety of studies on them to basically figure out what these biocatalysts do, and we can assemble this into a larger picture of how these metabolic pathways work, which overall answers the question of how the organism deals with the weird sugars of the ocean. So our approach to this is actually started with sequencing new genomes, just partly because I was interested in doing it, sequencing genomes, but also because there weren't, at the time we started this, very many genomes of marine microbes. Now there's a whole lot of them, but that's beside the point. And so it all begins with local bioprospecting. And for those of you who know me, they might recognize this person here. This is my oldest daughter, Nat, who's now a microbiology student, and she's now working on this project of bioprospecting, so hunting for new microbes. And so the pipeline is actually pretty simple. We take samples from the beach, as is being done here. You enrich them, you isolate individual strains, then you confirm the phenotypes that you want, and then you do next-gen sequencing and annotation. And this gives you a place to start to mine the genome and start doing biochemistry on the enzymes that are encoded in that genome. So where we stand now, we've sequenced 21 new genomes that cover a variety of different genera. And so here are some examples of after one does some genome gazing. So I just chose 3 examples here, and I chose them because you can see one is a specialist, and I think Jan is on the line today. So this is actually one that he isolated, which was an alginate specialist. He also isolated this PS47, which you can see is more of a generalist. And one of the newer isolates is this Pseudoalteromonas U2A. These are now being speciated. This is a Phylogenia species, and this is a distincta species. So we have a variety of different organisms that range from very specific to general. And what I want to focus on today is Carrageenan metabolism. So what's carrageenan? The base structure of carrageenan is this disaccharide, which is beta-1,4 linked D-galactose residues. And in its very base form, one of those residues is a 3,6-N-hydro D-galactose. These, however, are linked together. So these disaccharide units are alpha-1,3 linked to give the repeating backbone. In some cases, though, this anhydro ring is busted open so that it's a regular galactose. And you'll see here that you have a variety of different sulfation levels. And so what you end up with carrageenan is a fairly large family of polysaccharides with slightly different physicochemical properties. The most common ones are kappa carrageenan, lambda carrageenan, and iota carrageenan, which you can see vary from 1, 2 to 3 sulfations. And the lambda carrageenan has the 3,6-N-hydro sugar broken open into a regular sulfated D-galactose monosaccharide. And so in terms of our isolates, these are all Pseudoalteromonas isolates. You can see that they have different growth profiles. Here we have growth on iota and kappa carrageenan, and we can sequence the genomes of these, and then we can start to do some transcriptomics. That's shown with these numbers above here, and that allows us to isolate which part of the genome is responsible or responding to the presence of iota or kappa carrageenan. And these 4 different species here have very similar polysaccharide utilization locus. We call it the carpool pull for Carrageenan pull. Now, at the time that we were doing this, a paper was published on the genome of Pseudoalteromonas carrageenivora, which has long been a model for Carrageenan metabolism. And of course, the main enzyme is this, our ortholog here is this GH16C or CGKA, which is one of the original carrageenanases. And so all of this adds up to you know, a nice genomic picture of what carrageenan metabolism looks like, and it allowed us to dig really deeply into the biochemistry of it. And this is briefly summarized here, where we can do a lot of biochemistry and structural biology on the enzymes, and then we can assemble the pathway. And in this case, there's 2 pieces. There's import and depolymerization into oligosaccharides, which involves mainly endo-acting enzymes, including an endosulfatase. And then we have this cycling with the other exoenzymes, which will depolymerize the oligosaccharides into monosaccharides. And if you're interested, this is the paper, but this is not the focus today. The focus today is actually on lambda carrageenan, because we had this one isolate called Pseudoalteromonas distincta U2A. I'll just refer to it as U2A through the rest of the talk, because the rest of it is a mouthful. And when we looked at its genome, we found right next to that iota carrageenan pull was this set of genes which looked like it might have something to do with lambda-carrageenan degradation. And again, comparing to the Pseudoalteromonas carragenivora genome and the paper that was published on it, right next to its kappa-carrageenan pull, but in a different arrangement, was the same organization of genes. And there's a few tip-offs about why this might be active on lambda-carrageenan. But the point is that these genes were actually absent in the other Pseudoalteromonas strains. And that was one of the real tip-offs that this might target lambda-carrageenan, because this distincta strain could only grow, was the only one that grew on lambda. So this gave us an opportunity to try and understand how this extraordinarily sulfated polysaccharide might be utilized by microbes. So just due warning here, strap in, we're a structural biology lab. You're going to see a lot of structures as we go through here. And one of the reasons is that, and not only are we a structural biology lab, but for the sulfatases in particular, we find that structure tends to be an extremely useful tool to tease out what their functions are. And I don't want to go into details why, but just suffice it to say that's why you see a lot of structures in this particular study. So this all started actually by just growing up U2A on lambda-carrageenan and then just looking for some activity in the cellular fractions. And so this is a C-page gel where we have the culture supernatant, which has been incubated with some lambda-carrageenan, and then we have the intracellular fraction, which is basically just busted open cells incubated with the lambda-carrageenan. And you can see that there's depolymerization of this lambda-carrageenan. And so the next question became, well, where do we start with the pathway? And fortunately, from a paper that's about 14, 15 years old now, these GH150s had been identified as endo-lambda-carrageenanases. And so these were the obvious candidates for the enzymes that would initially depolymerize lambda-carrageenan. And so indeed, recombinant versions of these two GH150s did depolymerize lambda-carrageenan, but not the other carrageenans. And if you compare the pattern between what we see with the recombinant enzyme and this extracellular fraction, they look remarkably similar, although the gels are run under slightly different conditions. But what this boils down to is that in this lambda pull, that we call it, the GH150s are likely endo-acting. We can tell by the laddering effect. One or both is possibly extracellular, considering that we got activity in the culture supernatant. But the end result is that one or both of these enzymes work together to depolymerize lambda-carrageenan into a series of oligosaccharides. And here's an example. There'd be an even numbered oligosaccharides with the non-reducing terminus being an alpha-linked galactose, and then here we have the sulfation pattern on there as well. So to us, the real question at this point was, how do oligosaccharides get broken down to be able to yield the monosaccharides that the microbe could eventually use? And so we started off with these GH110s, and that's because the only characterized examples of GH110s are actually alpha galactosidases. So they seemed logical candidates to be able to take off that terminal sugar. And GH110B, GH110A, that's what we've called them. GH110B was easy to find an activity for. It was active on gal alpha 1,3 gal, which you should recognize as a repeating unit in lambda-carrageenan. It was active on the synthetic substrate, whereas GH110A was not active on any of these, which was a little bit disappointing, but you'll see why in just a moment. So through the structure of GH110B, and in this case a mutant in complex with gal alpha 1,3 gal, it was actually quite illuminating. And this is the -1 subsite, and you can see that the non-reducing terminus of this sugar, or extrapolate to a little oligosaccharide, is jammed right in the bottom of this active site. And this is the bond that would be cleaved. This is O6, which would be sulfated in an unmodified oligosaccharide, and it's actually tucked in there quite nicely. O2 is really jammed in the bottom of there, so this cannot be sulfated at all. We followed this up by solving the structure of GH110A, and from the native structure realized that there was a larger pocket surrounding where O6 would be in this particular enzyme. And then we determined the structure of this in complex with galactose-6 sulfate. And the architecture of the active site is very similar. It's cut away a little bit more, but it's still, this monosaccharide is jammed right down in there, but it can accommodate this sulfate. The O2 is still facing into the protein and cannot accommodate a sulfate there. So what this boiled down to is any exo-alpha-galactosidase activity must be dependent on sulfatase activity. I've just provided this here as a reminder that in this case, at the very least, the 2-sulfate has to be removed, and depending on the enzyme, the 6-sulfate must be removed. And so at this point, we went, okay, well, let's start looking at the sulfatases, because these are going to be key enzymes in this whole cascade. And there's 5 sulfatases in this pool, so it's really rich in sulfatases. And we actually went to NMR as one of the early assays, and we did not see any endo activity on lambda carrageenan by NMR. And so this was actually in a way a little bit disappointing, because we had no clues at this point. And so we ended up going on the hunt for the Exo-2 Exo-B-D-galactosidase, which I've called here the key to everything, and you'll kind of see why this is the key to everything as we go through here. And so we started off with a fairly simple approach, and that was to depolymerize lambda carrageenan with GH150B, and then add into that reaction one of the sulfatases, and then run the samples out on C-page gels. And you can see here the only one where there's a slight change, there's a slight shift in the banding pattern with S18B. At this point, we hypothesized that maybe this could be a 2-sulfo-D-galactosulfatase. And so we tested this under the idea that if it was removing this 2-sulfate, it would actually make GH110 have access to this alpha-1,3 link bond here, because GH110A was the one that could accommodate that sulfate. And so we did the same assay and just added in GH110A to all of these samples. And you can see here indeed that you get a profound change in the banding pattern, big shifts in the banding pattern. So at the very least, it looked like this sulfatase was making this oligosaccharide then susceptible to GH110A. We corroborated this by looking for released galactose using an assay that was actually sensitive to galactose 6-sulfate, fortunately. And you can see that only when you have the GH150, 110, and S18B do you actually get some free galactose released. So it certainly looked like the S18B was making this oligosaccharide, the products of GH150, be susceptible to GH110A activity. And so enter into structural biology. And most of the structures I'll show you today are actually mutationally inactivated enzymes, so that we can trap intact substrate complexes. And in this case, it worked beautifully. We could solve the structure of S18B in complex with an intact oligosaccharide. There's the active site there, which is zoomed in here, and you can see the 2-sulfate here engages the catalytic machinery in the active site exactly how you'd expect for a sulfatase. When we zoom in on the surface of the active site, you can see that the non-reducing end of this sugar, so this residue here, is tucked right in the bottom of an active site. 2-sulfate back there, 6-sulfate sticking out. Here is the alpha-1,3 bond, and I call this an alpha kink because, of course, this is an axial bond here, so it allows this sugar to point out of the active site, and it can access this oligosaccharide. This will become important in a little bit. So the activity data and the structural data clearly indicate S18B to be the Exo-2 2-sulfo-D-galactosulfatase that's active on this terminal G2 success. So with this, we could then generate this substrate, which should be the substrate of GH110A, and we solved the structure of a complex of GH110A catalytic mutant to trap a Michaelis complex, and indeed we managed to. This part of the active site, the -1 subsite, it's again the same as what I showed you in an earlier slide. O2 cannot be sulfated, it's tucked in there, the success can be accommodated. Here would be the bond that's cleaved. And then we have a variety of interactions through the +1, +2 subsites. In particular, you have a sulfate that's accommodated here in a basic pocket. So this is all adding up to these 2 enzymes working in tandem, the S18B and the GH110A. So if the S18B enzyme is a 2-sulfo-D-galactosulfatase, it then followed that the other 2 S18 family members would also be 2-sulfo-D-galactosidase. And it was just a matter of trying to figure out which one did what exactly. And so with this combination of the two enzymes now that I've showed you in the previous 3 or 4 slides, we had a way to get to this particular oligosaccharide, which terminates in a beta-link galactose with the 2-sulfate. In other words, it's the original oligosaccharide with this N monosaccharide removed. And we solved the structure of S18C in complex with this. We could only model 2 sugars, but that was totally fine. And what you can see is that the oligosaccharide again points down into a pocket active site, so we can't have extension on the end of this. It is another exo-acting sulfatase. The 2-sulfate is exactly where it should be for catalysis. Now, the difference with this, even though it's related to S18B in terms of the subfamily and sequence and all that, the contouring of the active site is slightly different in that it has to recognize a beta-linked sugar. If this were alpha-linked, that would be coming out there and the sugar would be banging into the surface of the active site. So there's some alterations in the contouring of this active site so that it's very specific to that motif on the beta-linked end, in contrast with S18B, which is contoured for the alpha-linked oligosaccharide. That left the mysterious S18A, and this is actually a structure that is in progress. It seems that we might have trapped potentially a covalent intermediate, but where we're at now is that we can model in a galactose quite clearly, and the active site is fully enclosed. And this would be 2-sulfated, engaging this region here as the catalytic site as we would expect. And it's fully enclosed mainly by virtue of this blue loop that I've shown here, which locks over the active site. And so what we have then is these other 2 S18 sulfatases. We have one that's specific for the beta-linked G2S, and then another one that's specific for the G2S monosaccharide. Initially, this was a mystery why this pull would even have this, but you'll see when we get to the end that there's actually a purpose for this particular weird enzyme. So where we are at here then is we have this beta-linked galactose at the end of our oligosaccharide that we've had some activity on by a sulfatase and GH210A and possibly S18C. And so how does that beta-linked galactose get cleaved? And well, the obvious answer here is this family 2 glycoside hydrolase. You know, the classic example of this family is LAC-Z, which is a beta-galactosidase. And so it turns out that GH2 is in fact active on PNP beta-D-galactopyranoside. And so we decided that we would pursue the activity of this on something that's more biologically relevant than a generic beta-galactosidase substrate. So the possibilities for this particular beta-galactosidase would be, is it active on the beta-linked 2-sulfated galactose or the naked galactose? And we turned to this galactose release assay to probe this. And just to remind you, this mix is actually the enzyme that depolymerizes the lambda carrageenan. This removes the 2-sulfate off the non-reducing end sugar, and then GH210A removes that. So this mix gets us to this point here. Then we added in either S18C, so that removes the 2-sulfate from the beta-link galactose to give us the naked end, or we added GH2, or we added both. And you can see when you add both, you get a profound increase in the release of galactose, indicating that the GH2 is actually active on this naked galactose here. And we could confirm this through some structural studies. We solved the structure of the GH2 in complex with the galactoisophagamine inhibitor. Not optimal, but it was the best we could do with this particular one. And I've shown you the surface of the active site here, and I've labeled these what they would be if this is a galactose. So of course, this would be the 2-hydroxyl group out here, and there's room for a 2-hydroxyl, but there's no room in this active site for sulfates anywhere, which supports this idea that this particular enzyme is removing the naked galactose, the naked beta-linked galactose. So by way of a quick recap, where we are at now is actually at a complete cycle for depolymerization. So here we have the ability to actively generate oligosaccharides from landed carrageenan by the 150s. We remove the 2-sulfate to generate substrate for GH110A. It removes this galactose-6-sulfate. We then remove the 2-sulfate from the beta-linked end to generate the naked galactose. GH2 removes that, and then we're back at the same end here. So you can imagine for any length of oligosaccharide, this going round and round and round to release galactose-6-sulfate, galactose, and sulfur. Okay. But then that raises the question, so what about these S115s? There's actually 2 more sulfatases in there. What do those do? And I mentioned GH110B already. So what does it do? Well, the obvious hypothesis was that if we have a series of sulfatases that are active on the 2 sulfates, well, these are probably active on 6-sulfate. And indeed, this was the case. So again, an inactive sulfatase that we solved in complex with galactose-6-sulfate. And in this case, we were able to model the beta anomer. And this is significant because this active site, again, is fully enclosed. We have this relevant tryptophan residue here, which starts to close in over the top of the active site. And the reason this is relevant is that this couldn't be an alpha. Now, remember, the only place a 6-sulfate is present is on the alpha-linked galactoses. So this can't be active on an oligosaccharide, no matter how you look at it, really. So this thing is active on free galactose-6-sulfate. And again, this will come in a bit, but you probably already noted that the GH110A enzyme is actually releasing free galactose-6-sulfate. So this particular sulfatase provides a way of converting this into free galactose. We then have, you can follow this, we then have the ability to make this particular oligosaccharide where the 2-sulfate is removed. So this is that initial S18B, the key to everything. And we solved the structure of S115B in complex with an oligosaccharide with that 2-sulfate removed. You can see the trisaccharide model in here. The structure of these enzymes is extraordinarily similar. Okay, the folds are nearly identical. The amino acid sequence identity is reasonably high. But the reason I point this out is there is a loop in the active site that is near the active site that's contoured slightly differently. So here's that oligosaccharide in there, and it pokes down. Again, we have another example of where we have this blind active site where you can only have recognition of the non-reducing end. We have the oligosaccharide protruding out. We have this galactose-6-sulfate here, which engages the catalytic machinery exactly as you'd expect. So what's the difference between these 2 enzymes? Well, it's in this particular loop that's near the active site. So what's shown in blue here is S115A. So that's galactose-6-sulfate. There's that tryptophan over here, which is closed in on this, this galactose-6-sulfate. In S115B, the loop is shorter, and so it's pulled away from the active site. This tryptophan here is analogous to this tryptophan. It's sequence conserved, so it's that one there. Okay, but it's pulled away, and this allows the active site to accommodate the end of an oligosaccharide. So while they're really similar enzymes, the slight shift in the active site makes one specific for a monosaccharide and the one be able to accommodate an oligosaccharide. And so with the discovery of this S115B, which removes the 6-sulfate, it gives this naked galactose, alpha-linked galactose, which is the substrate of the GH110B that I mentioned early on in the talk. And we could solve the structure of the complex here. And it is, as you would expect, very similar to that gal alpha-1,3 gal Michaelis complex. Here we have the -1 subsite, can't accommodate sulfates in there. And then we have this basic pocket, which is very similar to the one I showed you in GH110A. So the end of that story is that GH110B seems to be specific to hydrolyzing a naked terminal alpha-linked galactose. So now we have this description of all of the activity of the hydrolases in this pool. So let's start to put it together. So here we have our extracellular lambda carrageenan. We have our GH150s that participate in depolymerizing this to oligosaccharides. Now, where I've placed these, we haven't confirmed this based on bioinformatics predictions, though we think this is probably reasonably reliable, where we have this one, which is lipid anchored, and we have the GH150B, which is freeing the periplasm. We get a series of these oligosaccharides. We then have the key to everything. This all starts with the removal of that 2-sulfate on the oligosaccharide. And then we're going to have GH110A, which removes that 6-sulfated terminal alpha-link galactose. That is desulfated by this particular sulfatase to give free galactose. We have an alternative way of getting there, though. We can take off the 6S sulfate as well, and then have GH110B remove the naked alpha-link galactose, but we end up at the same place here. We then have this 2-sulfo-D-galactosulfatase, which removes that 2 sulfates to give the naked beta-link galactose. The GH2 removes that, releasing free galactose. Now, again, we're back at this particular motif: lather, rinse, repeat, and you can depolymerize the entire set of oligosaccharides. And then this is where that weird S18A comes in. If you follow this through, at the very end of depolymerizing any lambda oligosaccharide by this mechanism, you end up with a free 2-sulfate, a single free 2-sulfated galactose. And that's where this S18A, we think, comes in, is to desulfate that to give free galactose. You can then, the organism can then import this into the cytoplasm, free galactose. It can use that as an energy source, and it's good to go. So this is the final model that we've built through all these structural and functional studies. And so by way of summary, what we have here in these marine pseudoalteromonads is this quite complicated carpel. You can have this segment here, which gives the ability to metabolize kappa iota carrageenan, and then an add-on, this add-on module that allows the microbe to grow on lambda carrageenan. Now, the lambda carrageenan depolymerization relies largely on what I call an exo mode of action, and that is, once you get the oligosaccharides, it's just nibbling from the ends. And that's slightly different than, say, iota carrageenan degradation or some other algal polysaccharide degradation. Finally, we have this key to everything. This is a keystone enzyme without S18B, and we've showed this in linked assays that if you do not have S118B present, sorry, that should be S18B present, the cascade fails. So with that, I'd like to thank the major players, and there will be so many people who acknowledge that I'm just focusing on the lambda carpool. And so Dr. Andrew Heddle and Dr. Chelsea Vickers were the real drivers of the data that I showed you. And then we have other people that made various contributions over the years. And I would just like to finish with this particular slide because Andrew has got himself real employment. He got a proper job, leaving an opening in this particular area of research. So anybody who's interested in this can feel free to contact me. And with that, I'd like to thank you for your attention and take any questions.
Thank you, Al, for an excellent talk. Something tells me you have to be an expert in jigsaw puzzles. I mean, where you just puts these pieces together. It's very nice. So now the floor is open for some questions. You can either raise your hand or take it into the Q&A, and we'll guide it to Al. Jan Henrik, if you care, you can start.
Hello, Al. This was a very nice talk. Thank you.
Thank you, Jan. Nice to see you.
Yeah, it's really nice to see you too. So I have very short, but I have actually three questions, and they build on each other a little bit. Do these enzymes need salt?
Oh, I'd have to defer to Andrew. I do believe that when they purify them and they're assaying them, there is salt present. I'm not too sure if the requirement, can you still hear me?
Yeah.
Oh yeah, you froze for a second there. I'm not too sure if it's just that the enzymes need salt or it's just they're not happy in the absence of salt, which, as you know, tends to happen with these algal polysaccharide active enzymes.
Yeah, because I think many that act on these sulfated ones need salt, right? Or they are unhappy. And then I wonder, do you think if we put really highly sulfated polysaccharides from algae and we bury it on land, Do you think that the microorganisms can still degrade it because there's no salt?
You know, based on how complex those poles are, I would be surprised if there was any land microbes other than ones in the gut that would degrade these. Of course, I'm happy to be proven wrong.
So maybe this could be a really interesting carbon sink, but would probably be very expensive. Yeah. So and then the last question is, why do these marine polysaccharides? Why do you think, you have certainly thought more about this now, why do they need these sulfate groups? Because when there's like homolog structures in terrestrial system, we do not see so much sulfation. What do you think now about this? Why do they have so much sulfation in the ocean?
Yeah, well, I mean, it probably has to do with the physicochemical properties, right? I mean, take cellulose, which has zero sulfation. If it was sulfated, it wouldn't pack together and form that rigid structure, right? Which is important to what cellulose does. On the other hand, in the marine environment, I don't think they necessarily need something as rigid as cellulose. The algae probably don't want that. And so it makes the polysaccharides a little more flexible. They don't pack together, they gel, right? Rather than form crystalline matrices. And so it gives them some flexibility. So that's my feeling, is that it probably has more to do with the physicochemical properties of the polysaccharide than anything. But you know, I'm sure there's probably other people that have thoughts on that. You probably have your own thoughts on that as well, right?
Yeah, but I don't really know why this is. I don't know. But thank you for this.
Yeah, no problem.
Okay, then Bernard has a question for you, Al. Come on, Bernard, you can speak.
Well, I just put the question in the question and answer panel, but from what I understood, Al, hi, Al, good to see you.
Hi, Bernie.
From what I understood, GH110B and GH2 seem to have redundant beta-galactosidase function. Is that so? Or do they?
No, sorry, I probably went through it so fast that was missed. So GH110B is an alpha-1,3 galactosidase. It's just active on the naked terminal sugar.
Okay,
GH110B is the beta-galactosidase that's naked, active on the naked beta-linked sugar.
Right, and GH2 is also doing the same job, right?
Nope, GH110B and GH2, alpha versus beta.
Oh, sorry. Yeah, okay.
Yeah.
Okay. So I just wonder, why are the enzymes so specific when they act in the periplasm, where there is no competition anymore?
Oh, you mean whether they want a sulfate there or not?
Right.
Yeah, that's a really good question, and something that puzzled us, because we actually think that GH110B, which is the one that I presented to you as being active on the naked alpha-linked terminal galactose, we think it does have some low activity when the 6-sulfate is there as well. So clearly it seems possible that you could have a GH110 enzyme that is active on any terminal alpha-linked galactose, whether it's sulfated or not. So why there's this duplication to make them quite specific, I don't know. Chance. I know that's a lame answer.
Yeah, maybe because they need to be.
Maybe.
Yeah, they need to be, because, you know, look at your sulfatases there. It's clear that they need to be very specific, you know, because if not, then the alignment of the substrate with the catalytic machinery may be just too wobbly, not good enough.
Yep, possibly. Yep.
Then we welcome Mar to the arena. You can ask a question, Mar. You can speak up. You'll have to just unmute yourself, Mar, and then you can speak.
So thank you very much, Al, for a fantastic talk. And it's really nice to see you also. So my question to you is, you haven't shown any the biochemistry you have done, the incubations. You haven't done any quantitative work on this, I reckon. Otherwise, you would have shown it, right?
Correct.
My question is, can you, from the incubation times and from the concentrations of the enzymes, give any estimate for the turnover of the sulfatases? Because they seem to be very heavily, the activities seem to be very heavily dependent on each other, and the sulfatases are obviously the limiting step in the degradation. So do you think the sulfatases are like fine-tuned turnover-wise so that this works in a machinery, or are they slower, or what's your impression from the incubations?
Okay, this is a really fantastic question, and it highlights one of the challenges with working with sulfatases, which is why I had that strap-in structures ahead slide and mentioned that there's reasons why we prefer to go with structure. One of the big issues with sulfatases, particularly these ones from marine microbes, is getting them to mature into a catalytically active form. So the nucleophile in these enzymes is a formal glycine residue. So it's a post-translational modification where in these particular enzymes, it's a cysteine that's converted to the formal glycine. And so it's really difficult to get that to be quantitatively converted into the formal glycine when you produce these in a recombinant system. So we have done kinetics on sulfatases before, but the issue is that we never know whether we have 0.1% of it matured or 100% of it matured. We even tried mass spec to quantify this, but that failed miserably. So that is one of the first major challenges. Is it's not like glycoside hydrolase where we typically assume that we have 100% of it active. The other challenge is the substrate itself. So we have been using purified substrates. I put that in quotes because it's really partially purified. You can't buy these off the shelf, where it's a commercial product where there's been lots of QC and things like that. We do our very best to generate the best substrates that we can, but we're not always 110% sure of what that substrate actually is. So that's kind of the second, and getting them in quantity as well to be able to do kinetics is also difficult. So in answer to your question, we'd love to know exactly what you're asking, but it's something that we just haven't been able to tackle for those reasons that I outlined.
Thank you very much.
Yeah.
Thank you, Mar. That's a very good question. That was actually the one I was saving for myself, but you asked it better than I would, so appreciate that. Now we're heading towards, I'm sorry?
We have one more question here from
one
Nico.
more question, yeah, okay. Yeah, okay, Nico, go ahead, you can ask just a little bit and we'll get you on sound as well. We'll see if we can.
I can read the question if there's.
Yep. Sure.
So the question for Nico is what are these organisms doing with all the released sulfates? 3 sulfates for 2 galactoses. Yeah, good question. They have sulfate assimilation pathways. I'm not an expert in it, but there's a whole other set of pathways that deal with the sulfate. Precisely what ends up happening with it, I'm not sure, but it's not ignored. It's not just dumped out. It's actually modified. Okay, and so we have another one here. Do you have any idea of the distribution of in marine microbiomes? Microbiomes specifically, we haven't started to get into that yet. That would be fun. And in fact, a friend of mine, Wade, the other day was emailing with me and said, have you started doing metagenomics yet? And I was like, oh, I just don't think I can handle that at the moment. We're just we're doing targeted studies of polysaccharides. But what I can tell you is when we did targeted studies to generically pull out seaweed polysaccharide degrading organisms, we got a lot of carrageenan degraders. Now that might be just because of the environment that we're working in and possibly even the time of year, but it seems like a lot of them do have the capacity to degrade carrageenan. When you start probing the databases of marine microbes, the same pull comes up quite often in Pseudoalteromonas, and that paper by Gobet and et al. That I cited early on in the talk, they did a really good job of probing the databases and identifying where it is. So I would encourage, if you're really interested in that, I would encourage you to go look at that.
Thanks, Sabina, and thank you, Al.